Hello everyone. Today we will discuss about plant taxonomy, its introduction and its historical background, and we will discuss about the technique which is used for the preparation of herbarium. So let's start. Firstly, taxonomy. So it is derived from Greek word taxis, which means arrangement, and nomous means law or rule. So means arrangement by rules or lawful arrangement of living organisms, especially plants. Why it is important? Because it helps in proper and easy handling of vast number of plants in a suitable process of following certain principles or rules. So these plants are classified on the basis of certain rules and principles. And this term taxonomy was introduced first by A.P.D. Candolle in 1813 in his book Theorie Elementaire de la Botanique. So this is the publication. So this term taxonomy was first used by A.P.D. Candolle, that is Augustin Pyramus de Candolle. Then there was a scientist known as Lawrence, or taxonomist Lawrence, who defined taxonomy as a science which includes identification, classification, and nomenclature of the plants. Next, there is the important term taxon. It was first used by Adolf Meyer in 1926, a German biologist, for the animal groups. The term taxon indicates taxonomic group like variety, species और genus और higher group. So ये जो term है taxon, ये use की जाती है किसी particular species और particular genus और particular higher group के लिए. For example, we have species, genus. So these group represents the taxon. These higher groups also represents the taxon. So named group of organism like phylum, genus, or species is known as taxon. Then there was other philosopher known as Theophrastus, and he has written one book, that is Enquiry into Plants, and he attempted to arrange the plants into several groups, and is also considered as the father of botany. So this is the book of Theophrastus, that is Enquiry into Plants by Theophrastus, who is the father of botany. Then there was another taxonomist, Andrea Cisalpino. He also classified plants on the basis of certain characters, like on the basis of their habit. Habit के बेस पे इन्होंने plants को classify किया था into trees, shrubs and herbs. But he also took into account the characters of इन्होंने ovary के character को, fruit के character को और seed के character को भी use किया था for the classification of plants. The next there was Pliny. He compiled a monumental work entitled as Historia Naturalis. इन्होंने book लिखी है Historia Naturalis. So this is the book which was written by Pliny Secundii. Then there was another person named as Gaspard Bauhin. He has formulated the idea of genus. He gave the idea of genus in many cases and gave binary nomenclature to his plants, or he used the two names, that is generic or specific names for the plants. Then there was another person named as Joseph Piton de Tonfoot. He divided plants into two groups as trees and herbs. Trees or herb में इन्होंने divide किया था plants को, and he used the character of inflorescence for his classification, mainly inflorescence and flower for his classification. He was also credited for the concept of genus. He gave a clear concept of genus. So this is the portrait of Joseph Piton de Tonfoot. Next is important taxonomist named as Carolus Linnaeus. He played a very important role in this system of classification. He was a Swedish naturalist, proposed a system of classification which was published in one book that is known as Systema Naturae in 1735. In his system of classification, he focused on the character of stamens for classification. स्टेमन्स के कैरेक्टर को इन्होंने ज़्यादा कंसिडर किया है क्लासिफिकेशन के सिस्टम के लिए, और इन्होंने प्लांट किंगडम को डिवाइड किया है 20 क्लासेस में. He also used the number and nature of carpels for the system of classification. He distinguished the orders, that is the subdivision of his classes into further subparts, that is orders, on the basis of carpel, और कार्पल के नंबर और नेचर के बेसिस पे इन्होंने क्लासिफाई किया है ऑर्डर्स को. So this was the book written by Carolus Linnaeus, that is Systema Naturae. And there was another book which was written by Carolus Linnaeus, that was Species Plantarum, and the first edition of this book came out in 1753, which contained enumeration of all the plants known to him till date. इन्होंने इन सभी plants की डिटेल्स इस बुक में दी थी. So this is the book written by Carolus Linnaeus, and that is Species Plantarum. For his this great contribution in the field of taxonomic work, he was considered as father of modern botany and father of taxonomy. And there was another taxonomist named as Hermenegild Sentapu. He is considered as father of Indian taxonomy. This is the photograph of Sentapu, father of Indian taxonomy. Next comes fundamental components of taxonomy. So there are 5 main components of taxonomy. First one is identification. Identification means to know the unknown plants by comparing certain characteristic features with already identified plants given in the keys of identification. So how we can identify किसी भी unknown plants को हम कैसे identify कर सकते हैं? So that process is known as identification. For this we use certain written literature which are commonly known as keys, जिनकी help से हम plants को classify करते हैं. Then next is classification. It is the arrangement of plants into groups or subgroups according to a particular system of classification and in accordance with the rules of the nomenclature. So एक particular classification का system follow करते हैं हम plants की grouping के लिए, which should be universally followed and which contains certain rules for the nomenclature. Then third component is nomenclature. It is the technique of giving scientific names to the unknown taxon or plants on the basis of certain rules and regulation of International Code of Botanical Nomenclature, which is commonly known as ICN. Then description. Description is the recording of maximum characters of taxon, plant parts, plant in a technical language. These characteristics on the basis of which taxon is identified called diagnostic features. जो वो characters हैं जिसके basis पे हम plants को identify करते हैं या classify करते हैं, so those characters are known as diagnostic features. And the last one is phylogeny. It is the evolutionary history of a group or species of an organism, which representing in the hypothetical way starting with an ancestral type relationship. So from which type of ancestor that plant arises. So evolutionary history of any organism or plant is known as phylogeny. So ये जो components हैं, they are the main components of taxonomy. Next comes aims of taxonomy. What are the main motives of taxonomy? So firstly, it helps in the preparation of proper pattern of classification of plants, how to classify the plants. Then it also established the correct method of identification, nomenclature and description of plants, how to classify the plants, how to identify the plants, and how to give the scientific names. Then it also helps in reconstructing the evolutionary history of plant kingdom. It also helps to arrange the closely related plants together. जो closely related plants हैं उनको एक साथ रखने में help करता है on the basis of similarities and differences. It work out the evolutionary process amongst the texta में जो plants हैं एक common group में plants कैसे evolve हुए हैं उसकी study में help करता है. It gives the complete information of some plants which are endangered or getting extinct. So, and also it helps in genetic variability and biodiversity study of the plants. It also gives the knowledge about collection, preservation of the plant species, how to collect and how to preserve the plant species. So it is also favored by plant taxonomy. Next comes levels of taxonomy. According to Turrell in 1938, there are various levels of taxonomy that we'll discuss one by one. First one is अल्फा taxonomy. जो अल्फा taxonomy है इसमें आता है collection and identification of organisms सिर्फ morphological features के basis पे. So it involves collection and identification of organism on the basis of external visible features leading to the compilation of floras. So floras are the descriptive inventories of plant of a particular area. Next comes beta taxonomy. जो beta taxonomy है, it includes classification on the basis of gross morphology, external characters, और इसके साथ it also includes some evidences from genetics, cytology, anatomy, palynology, embryology, and physiology. So it also take the data from these various branches of biology into the consideration for the classification of plants, which include genetics, cytology, anatomy, and palynology. Palynology is the study of pollen grains, embryology, and physiology. Next comes gamma taxonomy. It deals with various biological aspects of taxa, ranging from the study of intraspecific population to the study of speciation and evolutionary rates and trends. So it includes the relationship between within the species or in the population, and it also studies the speciation, which is the origin of new species from the pre-existing species, and evolutionary trends, how evolution is taking place. Next is omega taxonomy. It considers all the visible microscopic, ultramicroscopic observations and biochemical evidences for the perfect grouping of organisms. So it includes all these informations into the consideration for the classification of organisms. Then the last term is systematics. Systematics is the study of diversity of living organisms, especially plants, and their identification, naming, and classification. And it also considers the evolutionary history of the particular species. This is known as taxonomy. Next are some basic related terms. First one is neosystematics or biosystematics. So it is based on all the characters of organisms which are gathered through different fields, including processes by which they have been evolved and maintains. So ये जो ब्रांच है ये इंक्लूड करती है सारी की सारी इन्फॉर्मेशन जो कि डिफरेंट बायोलॉजी की फील्ड से ली जाती है फॉर द क्लासिफिकेशन, और हाउ दैट प्लांट और स्पीशीज इवॉल्व्ड, इवोल्यूशनरी कैसे बनी है वो. सो ये जो सारा डाटा है which is used for the classification system is known as neosystematics or biosystematics, and this concept was given by Julius Huxley in 1940. Next comes morphotaxonomy. Morpho means shape, जिसमें हम external morphological features को study करके classification करते हैं किसी भी living organisms की, उसको कहा जाता है morphotaxonomy. Next comes chemotaxonomy, in which we study certain specific chemicals to a particular plants which are used for the system of classification. The next is cytotaxonomy. So it is the classification of living organism on the basis of cytological studies. Then karyotaxonomy, which is based on the number, structure, and behavior of chromosomes. Then serototaxonomy. In this type of identification marker, we use antigen antibody reactions, which are the main key points for the classification of organisms. Next important term is holotaxonomy. So it includes all the aspects of taxonomy which are completed, assembled, and analyzed. So सभी तरह के जो information है उसको collect किया जाता है, then it is assembled and analyzed for the proper understanding of evolutionary history of an organism. Next comes numerical taxonomy or taximetrics. It includes numerical evaluation of similarities between two groups of an organism and ordering these groups into higher ranking taxa on the basis of similarities. So इसमें comparative study की जाती है on the basis of numerical calculations. We study similarities between the two groups and place them in the higher ranking of taxa on the basis of similarities. Next comes Adansonian taxonomy, which is also known as phonetics. So it is a branch of numerical taxonomy in which organisms are grouped into taxa on the basis of estimate of overall similarities. So एक estimation की आती है similarities की और उसके बेस पे हम organism को group करते हैं into taxas. Next comes cladistics. So it is a method of classification of organisms employing genealogies, which is also known as study of the future generations or alone in inferring the phylogenetic relationship. So इसमें हम study करते हैं offsprings की और parental histories of an organisms, जिनको study करके हम plants classify करते हैं. In Hindi we call it as वंशावली. So it is an important technique of classifying the organisms. Next is phylogenetics. It concerned with the reconstruction of evolutionary history of an organism. Next comes herbarium. So what is herbarium? Herbarium is a depository which include well-dried, well-preserved specimens for the botanical studies. So this is the herbarium. These are the herbarium sheets, which includes various plant specimens which are well-dried and preserved and are kept in a particular cupboard that we will discuss how it is prepared for the future studies. So what is herbarium? It is a depository of dried, well-preserved plant specimens collected from different areas and mounted on the appropriate sheets having specific size and are arranged according to latest system of classification. So this is known as herbarium. So these specimens may be whole plants. It includes mains with roots or upper aerial portion or plant parts. And these will be usually firstly dried and then mounted on the sheet. So it can include this whole plant या फिर जो एक छोटा सा plant का portion है उसको use कर सकते हैं हम herbarium बनाने में. But these plant parts should be dried and well preserved. So this art of making herbarium was firstly started by an Italian physicist, which is physicist known as Luca Ghini. He was the first person to initiate the art of making botanical gardens also. Herbarium making by pressing and sealing specimens on the sheets of paper. Again, this is the herbarium sheet in which we preserve certain plant parts, and we also include the plant parts which contain most of the characters like fruits, flowers, and any other plant parts which helps in identification of these plants. So this is the herbarium. Next, why we need herbarium? So it plays a very important role in various aspects. Firstly, it helps in study of various plants of a particular area, and it also contributes to the scientific studies. Then it helps in preserving the catalogs of seeds and herbarium plants. We have some information. हमारे पास एक information आती है किसी भी plant की, उसके plant parts की, seeds की, flowers की, fruits की. So by studying these characters, we can identify the plants. Next is it also helps in maintaining the plants which are at the merge of extinction. Verge of extinction, जो plants extinct होने वाले हैं उनकी study करने में help करता है. And it also helps in informing the students and public about these specimens. And it also informed the effect of global warming on the ecological system. So these are the various purposes of herbarium. So this is the cupboard-like structure which shows various files or folders that contains sheets, and in these sheets they contain the plant specimens which are arranged in a particular system of classification. Next comes types of herbarium. And Davis and Haywood, they have classified the herbaria into three categories, which are the national or major herbaria, then regional herbaria and local herbaria. National herbaria, which include the herbarium of national level, then regional herbaria, which include the region, which includes the herbarium of a particular region, and then local herbaria, which include the region of a particular college, university, or a local area. Next comes techniques of making herbarium, how herbarium is prepared. So it involves following steps. First one is collection. In this step, we collect the plant material from various locations. First, then second is pressing. It involves proper pressing of that material to make it dry. Third one is drying. Then it is the drying of that specimens by using certain newspapers, blotting sheets, or some artificial methods of drying like oven and open sunlight. Next comes poisoning. So to prevent the attack of insects, we will fumigate those specimens. Then mounting. These specimens are mounted on the specific herbarium sheets with the help of glue or either using some threads or by sewing machine. Then next comes labeling. Labeling of the plant specimens, and we write all the important information regarding that specimen. Next comes identification and filing of that specimen. Finally, the storage. So these are the various steps which are involved in the preparation of herbarium. The first step in herbarium preparation is collection. In this step, we collect the plant material from various places. और जो plant material होता है that should contain some unique features, like it should contain flowers and fruits. And if it is herbaceous, then we will uproot it with root system and other underground plant parts. So that helps in identification. So the plant specimen should bear flowers, fruits if present, and in case of herbaceous small plants, we will collect it with roots or other underground plant parts. A twig size that is more than about 25 centimeter with leaves and flowers act as an ideal material for the preparation of herbarium. So data should be recorded in the field notebook at the time of collection. So all the unique feature of that plant should be recorded in the field notebook at the time of collection while collecting that plant material. Soon after the specimen are collected, then they should be pressed in the field itself. So जब भी हम plant parts को collect करते हैं, उसके बाद उसकी they should be pressed to remove the water from that plant specimen, और उसको dry करना पड़ता है. So this is the sheet which shows various plant parts which are used for this collection. So we'll collect these plant parts और उसके बाद इसको dry करते हैं after pressing it. Then for collection we require certain tools and equipments. So collection material, we require certain tools and equipments, like we require cutter, then we also require herbarium sheet, digger, then scissor, vasculum. In addition to this, we require camera so that we can capture the photograph of that plant. Then we require field press, in which we can press the plant specimen. Then we require shoulder bag, which is used for bringing the plant material. Then we require some specimen tubes, जिसमें हम plants के कुछ एक special plant parts को collect करेंगे. For example, we can collect the flowers or seeds for study. Then we require pruning shear, like this cutter that is used for cutting the plant material. Then we require, this is the vasculum, and the role of this vasculum is, it is a made up of metallic box होता है ये, और इसका जो size होता है that is 50 ideal size is 50 into 30 into 15 centimeter, and this vasculum is used for keeping the plants immediately. जैसे हम plant specimen को collect करते हैं, इसके अंदर हम इसको रख देते हैं generally in the forest, and if it is too hot in the environment, then we have to sprinkle some water ताकि इसके अंदर जो temperature है वो normal हो जाए. So this is basically used for the collection of plant material, जो हम immediately इसके अंदर plant specimens को रखते हैं. The next we require knife. जो knife है that is required to protect ourselves from wild animals. Then we also require some hand lenses or magnifying glasses, which are used to study some microscopic or some characters which are not visible with the naked eye. Then we also require first aid kit, which should be kept along with proceeding on the botanical collection of these plant materials. So these are some materials which are required for the collection of plant material during the preparation of herbarium. Next step is pressing. So it involves this placement of specimens in the center of pressing sheets, or ये जो pressing sheets है, it can be the blotting sheets or the simple newspapers. Initially जब specimen fresh होता है, then we have to replace those sheets on the daily basis with new sheets. So these are some collecting pressing equipments. इसके अंदर हम plant specimens को press करते हैं और उसके बाद इसको tie करते हैं with the help of these thread-like structures. So if the specimens are large enough, अगर specimens बहुत ज़्यादा लंबे हैं, then we should bend these, giving them the shape of V, N or W, so that they can adjust it in the herbarium sheet of having standard size of 42 into 28 centimeter. So in this photograph you can see जो plant specimen है, अगर इसकी length ज़्यादा है, so इसको हम bend करते हैं in the form of N or M or W. So this is a V-shaped specimen. So the bundle should be uniform in thickness. जो हम pressing equipment में रखते हैं sheets को, वो bundle uniform होना चाहिए thickness में, in the middle and on the sides. And the specimen should be kept one above the other, और जो specimen है एक के ऊपर एक specimen रखा होना चाहिए for pressing. So this is again the equipment which is used for pressing the plant material. इसमें sponge भी है that will helps in absorbing the water from the plant specimen. Then it also contains newspaper or blotting sheets जो पानी को absorb करते हैं. Initially they are replaced on daily basis, then after that they can be replaced after 3 or 4 days. Next comes drying. This is a very important process during the preparation of herbarium. So after 2 or 3 changes of these blotting sheets, finally the specimen is kept in the blotting sheets and dried either by natural or artificial heating. So dry करने के लिए specimens को हम place करते हैं specimen को drying sheet के नीचे. Drying mat है जो पानी को absorb करता है, and it helps in pressing and drying of the specimen. Again, this is the equipment which is used for pressing or drying the plant material. Then we can change the blotting sheet daily for 6 to 10 days initially, depending upon the weather conditions. अगर weather conditions humid है, then we have to replace these blotting sheets daily. And it can be accelerated. This drying process can be speeded up or accelerated by the placing the specimen in open sunlight or in the drying chambers, commonly the ovens. So this is the oven. We can fix the temperature, और हम इसके अंदर plant specimen को रखेंगे, and it helps in drying of that plant specimens. In case of succulents, which means the fleshy plant parts, they cannot be preserved by this drying method. So ये जो plant parts होते हैं, they are preserved in either boiling water or either in some preservative like alcohol or strong formalin solution as such. So these fleshy plant parts cannot be dried by using this drying method. So it involves the wet preservation of these fleshy plant parts. Like this, we can see these certain specimens which are preserved in the preservatives like formalin or alcohol. So these are various plant specimens which are preserved in the liquid preservatives. Next comes poisoning. So it helps in killing the plant pests and prevent the formation of abscission layer. Mostly it is done by dipping the whole plant in a poisonous chemical which is known as mercury chloride. इसमें हम plant parts को dip करते हैं ताकि uniform killing हो जाए tissue की, and that prevents the abscission of certain plant parts for 15 to 20 seconds. Then we can also use some other chemicals like lauryl pentachlorophenate, that is abbreviated as LPCP, as a substitute for mercury chloride and formalin. So we can use these certain chemicals also for the poisoning process. So this is the sodium pentachlorophenate, which is used for killing the plant tissue. Next is fumigation. So fumigation is mostly done to kill the insects or pests जो plant parts को damage करते हैं. So this is done to killing the insects which often damage the plants. And it's the most common method to keep the most common method of fumigation we can say is keeping the naphthalene ball. जो naphthol balls होते हैं उनको हम मुस्तन बैग के अंदर कपबोर्ड्स में रखते हैं जो prevent करता है attack of insects. Then we can use certain volatile poisons like methyl bromide, carbon disulfide, and carbon tetrachloride for the fumigation. Sometimes we can use para-dichlorobenzene for the fumigation process, and we can put this para-dichlorobenzene slices or small pieces in the cloth bags जो कि cupboard के अंदर रखे जाते हैं to prevent the attack of insect. And the chemicals may also be sprinkled on the sheets or bundles of these plant specimens. So this is the process of fumigation. We can use these machines which are known as fumigators, and we can use certain chemicals to fumigate the cupboards to prevent the attack of insects. Next comes mounting and stitching. This is the step after when the specimen is fully dried. So we can mount it on the specific herbarium sheets having standard size of 28 into 42 centimeter. So the standard size of the herbarium sheet is 28 into 42 centimeters. And we can mount it with the help of using certain glues, or we can also use threads. So by sieving we can mount these specimens on the herbarium sheet. So usually made up of durable card sheets. जो herbarium sheets हैं वो durable card sheets से बनी होती हैं, and it is advisable to mount one specimen. एक ही specimen को हम एक sheet के ऊपर mount कर सकते हैं. And the dissected और जो loose parts होते हैं plant parts such as flowers or fruits or seeds, they are kept in the paper packets. उनको हम paper packet के साथ paste कर देते हैं sheet के ऊपर. So like this we can see this is the female cone of pinus. इसको भी हम इसी के ऊपर plant parts को mount करते हैं. Next step in herbarium preparation is labeling. So there should be proper labeling of the specimen. जो भी information है specimen की, that should be written in the label which is put on the right hand side of the herbarium sheet in the bottom of sheet. So labels are put on the right hand side of the sheet in the bottom, and the label size varies. In some books it is written as 3 into 5 centimeters, and in some it is written as 8 into 12 centimeter in size. And it should contains the following information like name of organization for which we have collected the specimen, then name of the family of that plant, then botanical name of the plant, then local name or vernacular name of that plant, then locality of collection, कहाँ से ये sample collect किया गया है वहाँ का नाम, then date of collection, then habitat of the plant, and field notes and collection number of that plant. In addition to this, there should be name of collector also. जिन्होंने ये specimen collect किया है उसका भी नाम उस label में होना चाहिए. For example, we have this sample of label, name, date, location, and other information, then common name, scientific name, the name of organization. So we can put this whole information in this label. Next comes identification. So character of the plants are studied carefully, then we will match it with the flora of the region, and we will identify that plant. So it is also compared with the earlier identified plants. उनको देख के इस plant part को हम identify करेंगे. Then finally we will label this plant specimen with the latest accepted name of that taxon. So this is the label which is put on the right hand side in the bottom of the sheet of herbarium. इसमें हम सारी information plant के बारे में लिखेंगे. The next step is filing of specimens. So herbarium sheets are sorted out on the basis of family, genus or species wise, or same species are filled in the covers which are commonly known as species cover, जिन्हें species cover कहा जाता है और folder कहा जाता है. or species belonging to the same genus or one genus are placed in the other folder, which is made up of heavy paper. This is called genus cover. And color of both the covers are different, that is the color of species and genus cover are different. So this is the folder, like for example this is for species, so we'll put all those herbarium sheets which belongs to the same species in this folder, and these folders are arranged in the cupboards on the basis of latest system of classification. Then the last step is proper storage of the plant specimens. So these plant specimens which have been properly mounted, identified are filled systematically in the special wooden or steel cabinets, and the herbarium sheets loaded with specimens are filled inside specific folders, जिनको species folder or genus folder cover कहा जाता है, and which indicates the proper species, genus, family, geographical area, etc. So ये ऐसे arrange किए जाते हैं in the wooden cabinets, and the plants are arranged and sorted following the Bentham and Hooker system of classification in case of Commonwealth nations. जो Commonwealth nations हैं वो इस system of classification को follow करते हैं. Then we can also follow one phylogenetic system of classification, that is Engler and parental system of classification, for the proper sorting out of these herbarium specimens. So these are some wooden cabinets in which we can put these herbarium sheets. So ये जो wooden और steel cabinets हैं इनमें इनको properly classification के basis पे arrange किया जाता है in the proper cabinets. Next comes roles of herbarium. So they act as a repository of dried plant specimens, and they helps in study of these specimens. Then there are several reputed herbaria in the world which keep the type specimens. So type specimens are those specimens which are described first time, जिनका description first time दिया जाता है, they are identified and they are named first time. These are known as type specimens. So they act as the principal proof of the existence of a species. Then these original documents upon which the knowledge of taxonomic characters rest, वो help करते हैं for the developing various floras, manuals and monographs. These are the documents which contain information regarding the particular species of the plant. Most herbaria have specimens collected from different parts of the world, and thus they helps in providing the information regarding the geographical distribution of the various plant species, जिससे हमें वर्ल्ड में different species का distribution का knowledge मिलता है. So these are various roles of herbarium. Next we have major herbaria of the world, which contains a list of specimens. Firstly we have the Museum National d'Histoire Naturelle. So this herbarium is located in Paris, France, और ये it contains 9.5 million plant specimens. Then we have New York Botanical Garden Herbarium. It is present in the Bronx, New York City in USA, and it contains 7.2 million plant specimens. Then we have Komarov Botanical Institute, which contains 7.1 million plant specimens. It is present in St. Petersburg, which is located in Russia. Then we have Royal Botanical Garden Kew Herbarium, which contains 7 million plant specimens. It is present in Kew, England. So this table shows a list of various world's herbaria which contains various number of plant specimens. Next we have important herbarium of India. So this table shows the various herbarium. For example, we have Central National Herbarium, which was established in 1795 by William Roxburgh, and it contains 2.5 million species, and it's located in Kolkata, West Bengal, India. Then we have other herbarium of FRI, that is Forest Research Institute, Indian Council of Forest Research and Education. It contains approximately 3.4 lakh specimens, which is located in Dehradun, Uttar Pradesh. Then we have Botanical Survey of India Southern Regions and Regional Center. It is located in Coimbatore, Tamil Nadu. It contains 2.5 lakhs specimens. So these are some enlisted herbarium which contains these number of plant specimens located in India. Then these are some references that I have used during the preparation of this presentation. So this was all about today's discussion. If you have any questions, queries and any suggestions, you can give it in comment section. Thank you. Have a nice day.